Dear colleagues, my name is Ekaterina Mironova. I am PhD, Head of Laboratory of Molecular Neuroimmunoendocrinology, St. Petersburg Research Institute of Physiopulmonology, and Senior Researcher of Laboratory of Molecular Mechanism of Aging, St. Petersburg Institute of Bioregulation and Gerontology. Today I will tell you about immunomodulating properties of peptides in vitro. The effectiveness of the immune system functions that ensures the maintenance of constant level of the body antigens decreases with age increasing. The process of endogenous and exogenous antigens recognition becomes less accurate. The intensity of immune reactions decreases. This leads to the appearance of immunopathological syndromes, immune deficiency, autoimmunity, increased levels of circulating immune complexes. Immune deficiency manifests in cellular and humoral immune response decreasing. Antigens are substances that penetrate into the body, are capable of stimulating an immune response, cellular reaction, antibody formation, allergy tolerance, and specifically react with the antibodies formation. Immunomodulators are various biological activity substances that turn a pathologically altered immune response into a physiological norm. If there is inhibition of any part of the body immune system, the immunomodulatory substance should strengthen activated to the level of the physiological norm. In the case of the pathological increase in immune status indicators, the immunomodulatory substance should selectively suppress this part of the immune system to a physiological norm, for example, in autoimmune diseases. In this record, the search for new effective and safe medicines aimed to restoring the immune system functions is especially relevant. The study of the molecular mechanism of immune system regulation using peptides bioregulators opens up new prospects for the immunomodulatory medicines development that are highly effective at normalizing the immune system functions and free of side effects. From the polypeptide complex fladonix, which was isolated from the thymus gland of young animals, the tripeptide cristagen was synthesized. Based on the analysis of amino acid composition of fladonix, the dipeptides EW and KE were designed and then synthesized. It was found that these short peptides have a wide spectrum of action in different experimental models. It was found that fladonix and dipeptides KE and EW stimulated an exosome vesicle concentration increasing in the monocytes. This indicates the cell's mitogenic activity increasing under the influence of thymus medicines and the ability of peptides to increase intercellular communication, protein secretion, and the immune response. The use of fladonix and peptides KE and EW also contributed to the activated kinases phospho-ERK and phospho-GNK synthesis stimulation in differentiation THP1 cells macrophage phenotype. Influence of fladonix and its component dipeptides KE and EW indicates an increase in the ability of differentiated cells to neutralize the negative effects of oxidation and damage with caused by free radicals. This molecular mechanism of thymic preparation immunomodulatory effectiveness is an element of body detoxification and protection against cellular degeneration mechanism. Dipeptide KE is an inducer of pineal gland immune cells differentiation, which can play an important compensatory role in age-related atrophy of the immune system central organ, the thymus. Dipeptide Ke promoted the differentiation of lymphocytes precursor into T helper cells, cytotoxic T lymphocytes, and B cells. In relation to the thymus and spleen tissues of old rats, Christogen had a tissue-specific effect, manifested in stimulating the growth of these tissue explants. It was also found that Christogen significantly increased the proliferative activity of thymic epithelial cells during cell culture's aging, with the most pronounced effects in the seventh passage. The data obtained indicate the immunogeroprotective effects of the peptide. Thus, the presented studies made it possible to identify the molecular action mechanism of the thymic medicine Vladonix and its active components, the Christogen tripeptide and the dipeptides EW and KE. The immunomodulatory effects of Vladonix are due to the effect of the short peptides EW, KE, and EDP included in its composition. Thymic peptides increase the extracellular vesicles level in monocytes, which indicates a cell apoptosis decreasing, their proliferation and division stimulation. Peptides also activate cytoplasmic kinases, phospho-ERK and phospho-GNK in the macrophages. This increases the cell's antioxidant activity and opposes the free radicals damaging effects. Normalization of the protein synthesis that activates the immune and antioxidant system functions, as well as the regeneration and hemostasis processes, is manifested in the body regulatory system restoration. This also explains the high effectiveness of thymic peptides in the treatment of a wide range of diseases. Thank you for your attention.